Hello everyone. What we're going to do here very quickly before we start is we're going to have a look here in the notes copy, what you should have written down, or what you're going to write in between here and next Monday. So what we have got here is a bacterial cell, a simplified drawing. This is the one I want you to take down. On the outside we have the capsule. Next we have the cell wall. Next we have the cell membrane, and a few other parts. Then inside here we have got the red dots representing the ribosomes. We have the DNA. We have the plasmid, the flagella. So that is kind of really what you need when you're drawing a bacterial cell, and all of the parts and functions there are for you on the right. There's a few extra details here in this particular slide that we'll be going through today, particularly the different types of bacteria in terms of their feeding habits. But what we did cover yesterday was the different shapes of bacteria. There's three shapes: rod-shaped, spherical, spiral-shaped. And I suppose we also recognize that not all bacteria are harmful. Some of them are harmless and actually beneficial to us. This one here, I just want to go through this in a little bit more detail than yesterday. We mentioned this yesterday. I had a video up that didn't work. But just to highlight here that the first step here, we say a bacteria that's capable of division would be like a bacterial cell maybe that has been around for long enough, that it's big enough to actually reproduce. We call this cell or rather a mother cell. So we call it a mother cell. And in this mother cell, the DNA of the mother cell is going to replicate itself, and it's going to make an exact copy. What I have highlighted here is that at the same time, the cell has actually gotten longer. So we say the cell elongates because it just does, it gets bigger. And what we notice next then is that the DNA will move to opposite poles of the cell. So the black DNA representing here on the left and the red on the right. And when we say the opposite poles, that's just like a term that we use in terms of cell division for saying the opposite sides of the cell. So the next thing then that will happen is we form a furrow, a cleavage furrow. A cleavage furrow, I'm sure you've heard of the word cleavage before, but this cleavage furrow will deepen then, and the cells will eventually split apart, and then around these cells, the cell walls will actually reform. So reproduction for bacteria actually is very easy to remember. You start with a mother cell, the mother cell elongates, the DNA replicates, the DNA moves to opposite sides, a cleavage furrow develops, and the cells split apart. The one thing to notice about these cells here, that we call them daughter cells, and the reason we call them daughter cells is because they are genetically identical in this case when we're talking about bacteria. So you have a mother cell and two genetically identical daughter cells, which are themselves going to eventually reproduce themselves. The type of reproduction rather, it's asexual because it only involves one parent, but it's also called binary fission because that is the name given to bacterial reproduction. Bi meaning two, so you'll end up with two cells, and fission meaning splitting of, so the splitting happens here. Right, so that's enough on reproduction because I went over that yesterday. Now what I'd like to do is I'd continue a little bit more here with theory today on bacteria. It might be a good idea at this moment in time if you pause the video and see, can you answer these questions? All of the answers for this will be inside your notes, but maybe I will discuss number 4 now, right now. So all bacteria are harmful. It says discuss this in 2 to 3 sentences. Well, I have already mentioned here that not all bacteria are harmful. Some of them are perfectly harmless, and in fact, some of them are actually beneficial to us. So what we could say here is that there are bacteria out there that benefit us, like, for example, gut bacteria. Gut bacteria are present in our large intestine, and what they do is they produce vitamins B and K for us, which are essential vitamins in our body. You could also talk about bacteria's role, for example, in decomposition. So there's many bacteria out there that will decompose, we'll say, dead matter and return nutrients to the soil, and that's essential for things like the carbon cycle and also things like the nitrogen cycle. So without bacteria, those essential processes could not happen. So we know that not all bacteria are harmful. Okay, many of them are actually beneficial. So When we look here at, and I'm going to skip on from this, when we look at bacteria, bacteria are, I suppose, all around us. And bacteria are, I suppose, one of the original, I suppose, forms of life on our planet. Bacteria are capable of surviving in incredibly hostile conditions. Like, for example, we can find bacteria on the surface of, I suppose, wooden surfaces around us. We can find it in our homes. We can find them in the soil. We can find them in the air floating around us. They're on our skin, but they're also in, for example, volcanoes. They're found deep in the ocean, in highly pressurized environments. And bacteria, in many cases, are adapted to survive in these hostile conditions. So, one kind of strategy of bacteria is bacteria have the ability to develop what we call an endospore. An endospore is like a tough kind of outer coating here that will develop here around a bacterial cell when the cell is in danger. Now, the cell could be in danger because conditions suddenly change. Like, say, for example, now, let's take, for instance, where you take your food and you stick it into a freezer and you lower the temperature down significantly in a freezer. What could end up happening there is that the bacteria have come from an environment where maybe room temperature was 20 degrees Celsius, and suddenly now the temperature is so low that the bacteria will try to protect themselves from those harsh conditions by putting an tough outer coating around their cell to protect their DNA. And so this ability of bacteria allows them to survive in hostile conditions, as I mentioned, like, for example, living inside in hot springs, living inside in volcanoes, living inside in radioactive environments. When bacteria encounter highly hostile environments, they always develop an endospore. Now, an endospore, we say, is a resting stage of a bacterial cell. It's a resting stage because the bacterial cell reduces all of its metabolism, and it's really slowing down everything until such time as conditions return and become favorable. So bacterial cells, as we know, like, for example, there's resting stages of bacteria that survive, we'll say, on meat, for example. But if you leave meat out long enough on a warm surface, eventually these bacterial cells will take over the meat, and they'll start, I suppose, inhabiting much of the meat and cause it to go rotten. So bacteria have this unique ability as an endospore to get onto most surfaces, and that makes them really resilient, actually, trying to, I suppose, kill them completely. So what you'll find is many, I suppose, cleaning agents claim to kill maybe 99.9%. And that's because many of the other bacteria here are, I suppose, the remaining like 0.01% or 0.1%. What they're doing is they're protecting themselves with an endospore which cannot be broken down. So what we want to talk about next then, and I'm going to come back to the endospore because we'll see this when we look at the what we call the growth phases of bacteria. Bacteria, as I mentioned yesterday, are, I suppose, adapted to kind of live in most environments. They can live in volcanoes. They can live in the deep ocean and live off of, we'll say, volcanic vents underneath the ocean. They live in the rivers, the water around us, and they live on food. Like, there's so many different places that these bacteria survive in. So this really broad group of bacteria, what we're going to say about them is that they're so broad, really, that they're either autotrophic or they're heterotrophic. So there's a large amount of these bacteria that actually survive completely on their own because they make their own food. So they're autotrophic. There's two different types of autotrophic bacteria we encounter on our own planet. We encounter photosynthetic bacteria. So in our oceans, for example, if you've ever gone swimming in the ocean and you came across kind of green water, it's highly likely that they were photosynthetic bacteria in the water that you encountered. These are bacteria that are using sunlight to produce their own food. They're just like plants. They photosynthesize. There's also chemosynthetic bacteria. Chemosynthetic bacteria are organisms that will use chemical reactions to produce food, to produce energy. So, like for example, in the nitrogen cycle, we will encounter bacteria that are capable of changing one chemical to another chemical form, and in the process, use energy from themselves. So, all of these adaptations, of course, have happened over millions of years, and I suppose just like any other organism, they are designed to live in certain habitats. So the heterotrophic bacteria come under either saprophytic organisms or symbiotic organisms. When we say saprophytic, we mean that organisms feed on dead matter. So bacteria really are the decomposers, one of the main decomposers in nature. And that means when an organism dies, for example, if you see, for example, a fox or a badger or something, a bit of roadkill, then that will start decomposing pretty damn quickly, and that's because bacteria will quickly colonize the dead organism, and they'll start reproducing and then start feeding on that organism and converting those organic compounds into something smaller. We also have the symbiotic bacteria. And symbiotic bacteria are organisms that will live in close association with other organisms under three different relationships. So, bacteria could live in a relationship which is mutualistic, and that means that both organisms actually benefit from that relationship. A perfect example, actually, of that would be where we talk about gut bacteria. Gut bacteria, for example, in our large intestine, they produce vitamin B and K for us, as I said, but we also provide them with a habitat where they are living, and we provide them with food because they are breaking down things like fiber. So that would be a mutualistic relationship where both organisms benefit. We also have commensal relationships. Commensal relationships, I suppose, I'm not sure does the word commensal come from business terms, but a commensal relationship means that one organism will benefit and the other is not harmed. So, for example, if we talk about the bacteria that live on our skin, if we took a sample of skin and put it underneath the microscope, we would find that there are a lot of bacteria living on our skin. And those bacteria are benefiting because they have a habitat, but they're causing us no harm in the process. The third, I suppose, type of organism are usually the disease-causing bacteria, and they are the parasites. Parasites are organisms that cause harm to another organism, but they themselves benefit. So if you think about, for example, if we talk about streptococcus, streptococcus is the bacteria that invades your throat and causes you to have a sore throat. So the streptococcus is benefiting, of course, because it is producing, it is getting food for itself, but in the process, it is damaging the cells within your body. Like, just one interesting side fact for you, maybe, we believe in evolutionary terms that, like, the more, I suppose, the longer an organism lives on our planet, the more likely it is to try to develop into a commensal or a mutualistic relationship, because it has a longer, I suppose, chance of survival in the long term. So the parasites, we believe that many of these parasites may transition into becoming, I suppose, entering mutualistic or commensal relationships, because I suppose the last thing you want to do is cause your host to die, which therefore you'd lose your habitat. So, skipping on, we know that there are like an enormous amount of bacteria, and we know that bacteria are the most diverse group, the most numerous group on the planet. So there's a huge population of them. There's probably more than every other organism put together. And there's also a huge biodiversity of them. So there's so many different like species of bacteria out there. Right. So a few different questions here that are appearing on the screen. And again, what I'm going to ask you to do is pause the video and have a go at them. And what I will do then is I will shout out the answers. Okay, so I am going to call out those answers now. What is the function of a plasmid in bacteria? The plasmid is an organelle that carries antibiotic resistance for bacteria. How quickly does binary fission take place in the average bacteria? It takes place about every 20 minutes. What term is what term is given to the dormant stage of bacteria when a tick cell wall develops? We call this an endospore. What are the benefits of this dormant stage? Well, it can remain in a resting stage whilst conditions are unfavorable or damaging to the cell. It can then come out of hibernation and when conditions are favorable and when it can maximize its growth. Finish the chart below. There's autotrophic bacteria and there's also heterotrophic bacteria. Underneath heterotrophic, we will have saprophytic and we'd also have symbiotic. Underneath autotrophic, you'd have chemosynthetic and photosynthetic. The final question there, this question here states, what factors do you think will affect bacterial growth? And I already mentioned one in this video, and maybe just focus on that one for today, which is temperature. Temperature absolutely will affect the growth of bacteria because we know at high temperatures you might even kill bacteria. Like that's why we normally are advised to cook meat well in the oven because the high temperatures will kill the bacteria. And inversely, we could talk about using a refrigerator or freezer to slow the growth or actually kill the bacterial cells in the freezer due to the low temperatures. Right, the details of your homework will be posted on Google Classroom, so check that out there.